Hello, everyone.
Hello?
Welcome to an overview on seed germination. Today, we will be addressing some key questions such as what is seed germination? What signals initiate the process? And what does the process entail? Let's get started.
Sure.
We will first take a look at the seed itself. The seed is an embryonic plant protected by a seed coat. The seed coat covers the embryo, which will eventually develop into a seedling through germination. As this occurs, the other parts of the embryo will indicate their respective future structures. The cotyledon will develop into the first leaves. The hypocotyl will become the germinating seed stem, while the radicle will form the plant root. There are small variations in the seeds between different types of seed-bearing plants. For example, monocot seeds only have one cotyledon, while dicot plants have two cotyledons. Seems like the seeds have all the components needed for it to germinate. But what exactly is seed germination?
What is it?
Well, it is the first stage of plant development when a seed sprouts and begins to develop into a mature plant. Simple to understand, right?
Yeah.
Well, actually, not quite so simple.
Oh?
The process is fairly intensive and requires just the right conditions. These conditions include the seed to be healthy, moisture, warmth, nutrient-rich soil, and sunlight. You may be wondering, how do plants sense these conditions? Well, plants like to use receptors and signaling hormones. Let's take a quick look at them.
Okay.
Let's start with phytochrome. The pigment phytochrome absorbs light and has two forms, PR and PFR. PR is inactive. It absorbs red light at 660 nanometers and transforms into the metabolically active PFR. The latter promotes germination and other phytochrome-controlled processes in plants. PFR reverts back to PR after absorbing far red light at 730 nanometers. In the dark, PFR slowly changes back to PR. In general, red light stimulates seed germination, whereas far red light inhibits it. Light requirements for seed germination may be replaced by plant hormones.
Wow.
Several development processes of plants controlled by phytochrome may be mimicked by appropriate hormones. Speaking of hormones, we can begin to look at two critical hormones that can affect seed germination: gibberellic acid, or gibberellins, and abscisic acid. These two hormones work against each other in their effects. Abscisic acid, or ABA. Known as the dormancy-inducing hormone, ABA prevents germination, as well as promoting stomata closure. The concentration of ABA has been shown to increase during the onset of dormancy of the embryo during seed development. It inhibits gibberellins biosynthesis, which is needed for seed germination to proceed, as seen in the right side of the figure. Gibberellic acid, or gibberellins, or GA. On the other hand, GA will initiate germination, since it is vital in promoting food breakdown for the seed. GA will signal to the outer layer of the endosperm, known as the aileron, to produce and secrete hydrolyzing enzymes, such as amylases and proteases. These will break down stored food in the inner endosperm cells. Now that we have all the signals out of the way, we can now start going over the process.
Finally.
After the seed is planted or buried in the soil, it can prepare to germinate. Firstly, the seed will undergo imbibition. This is the absorption of water by the dry seed. Imbibition results in the swelling of the seeds. The swelling takes place with a great force, aka turgor pressure. This will rupture the seed coat and sprout the radicle. Imbibition rehydrates essential macromolecules, chiefly the cell wall and storage polysaccharides and proteins. Now that the seed has absorbed water, metabolic activity can resume and respiration begins. Initially, respiration may be anaerobic. Eventually, it will switch to aerobic respiration as the plant begins to take in oxygen. The seeds of water plants, such as rice, can germinate underwater by using dissolved oxygen, requiring less than land plants, who obtain their air contained in the soil. This is why seeds are loosely planted in the soil for land plants. Seeds planted deeper in the soil can often fail to germinate due to insufficient oxygen. At this point, light can really affect the seed germination process. The seeds which respond to light are known as photoblastic seeds. There are three types: positive photoblastic, negative photoblastic, and non-photoblastic. Positive photoblastic seeds require exposure to sunlight for germination. Negative photoblastic seeds, on the other hand, do not germinate if they're exposed to sunlight. Finally, non-photoblastic seeds germinate regardless of their exposure to sunlight. This ties in closely to our previously discussed phytochrome pigment, which is responsible for sensing light. As the seed continues to germinate, the embryo resumes metabolic activity, and cell division and expansion takes place. This metabolic activity uses up stored starch, proteins, and fats. These food reserves may be stored chiefly in the endosperm or in the cotyledon, varying between plants. These food solutions comprises of sugars and amino acids, which are diluted by water and pass through the cotyledon. From here, they're transferred into the epicotyl, hypocotyl, radicle, and plumal. Once again, this entire process is facilitated by GA signaling. Lastly, once the food is transported and utilized, the cells of the embryo in the growing regions can start up their metabolic processes. The cells in these regions will grow in size, undergoing cell division and developing the seed into a seedling, completing the germination process.
Any questions?